Okay, so if you remember yesterday, we were talking about protein synthesis and we covered the two steps of protein synthesis: transcription, the writing of the code from the DNA to the RNA, and then we talked about translation. Now that RNA code is read by the ribosomes, and the tRNA come in and bring the amino acids that are going to build those proteins or those polypeptides. And so, what we're going to look at today is the process of transcription and translation using DNA bases and working through transcription and translation. I want to introduce you to what we call a codon chart. This chart is one of many different versions on what a codon chart can look like. I prefer to start with this one to teach students because most of the time it's easier for them to understand. And so I will walk you through how we read this codon chart in just a minute. The codons have to do with the 4 bases that are found on the RNA strand that we're going to build from our original DNA strand. So we're going to start with DNA. Remember that we're coding for traits. We're making proteins or polypeptides that are coding for certain traits. So, for example, let's do eye color. Eye color has tons of traits. We call it a polygenic trait because it pulls from all these genes on all these chromosomes all throughout your DNA. But for the sake of keeping it simple, if we were going to try to code for our eye color and we're looking at all these genetics, we're going to start pulling the genes from the DNA strands that have to do with eye color only. And in order to build the proteins that are going to make the pigments for our eyes, we have to go through the process of protein synthesis. So at this point, hopefully you're following that we're pretending to be those protein builders. We're helping this process along the way. Here's our strand of DNA. This is the gene that we're pretending codes for eye color. Okay, so I have a list of nitrogen bases here. A, T, C, and G, they're all here. Now, if you remember when we were coding or base pairing DNA and DNA, we use the same 4 bases. But when we base pair DNA and RNA, there's something that changes. Every time you come across an A, you don't pair it with the T in RNA, you pair it with a U. So remember that the U replaces that T in an RNA strand, uracil. So we're going to base pair our DNA to our RNA first. That is the first step. This is where our little, and I'm going to put the M here, our messenger RNA comes in. This is the process of transcription. We are literally writing, we're scribing our RNA strand now. A pairs with U, C pairs with G, A with U, T still pairs with A, G pairs with C, C pairs with G again, A with U, C with G, A with U, and T with A. So now we have our messenger RNA coded. It's ready. Now this is happening inside the nucleus, don't forget. We're inside the nucleus, this is happening. Once this is written, it gets to move, and it's going to move out of the nucleus and into the cytoplasm, just like we looked at yesterday. Here's where things start to get built. Pretend for a moment that this has moved. Actually, what I'll do for you is I'll go ahead and rewrite this over here, just to make it easier for you. Let's put this. We have UG, U, ACG, GAU, GUA. And you're going to probably notice that I automatically set it in groups of 3. That's because my brain automatically sees it after doing this for a while. My brain sees it in sets of 3. And that's what I'm getting to in just a moment. I'm actually going to scoop this over a bit so you guys can see this side of the screen. There we go. Okay. As I was just mentioning, this RNA strand, it's the same strand that's here. I just moved it over so we can work with it. We look at this RNA strand and we break up the bases into groups of 3. And I'm going to do that right now. Okay, so in this particular gene, because all genes have different lengths, they have different sizes, different number of bases, I've got 1, 2, 3, 4 sets of 3. That's 12 bases. Each of these is a codon. Okay, so this is a codon, this is a codon, this one is, and this one is. These are important because this is how we're going to figure out what amino acids are going to be built to make that polypeptide or that protein. This is where the codon chart comes into play. So what we're going to do is we're going to look at this first codon, UGU, and we're going to find it on that codon chart. And this is how we do that. Your first step is to go to the center of this chart and find the first letter. U, C, A, and G. These are the 4 letters we work with. Our first letter is U. That's our first step. Now, once I'm in this section here, notice I've got 4 letters again. U, C, A, and G. So I've got U, G, and then U. So U, my second step is to come here to the G. Now, my last step is going to be to take the third letter, UGU, and I'm going to find it in this little section here, UGU. And so my third step tells me that I have cysteine as my amino acid, and that is abbreviated as CYS. And so all I'm going to do right now is I'm going to go in and I'm going to write CYS. That is the amino acid that I just built using this codon. Now, here's the part that you don't see when you're doing this. When this codon was read, there's a nice little tRNA that's carrying it. And it's carrying it with the matching anticodon. So what it is it's carrying something that matches this. U goes with A, G goes with C, and U goes with A. So actually this tRNA has this code on it. We call that an anticodon. Don't focus too much on that, but I want you to see how this works. This tRNA is carrying that amino acid and finding this codon to match with this anticodon. When it does, it drops that amino acid off, literally drops it off into the link that's going to become the polypeptide. And so this is going to happen with all of these codons. The tRNA is going to come in with an anticodon to match. That's going to build the amino acid. Okay, so now let's try the next one, going back to the steps here with this codon chart. Our next one is A, C, and G. So we start with first letter, second letter, third letter. We read them left to right. Find our first letter in the middle, A. Now we're only working in this quadrant here, A. Our second letter is C. Now we're only working in this section. And our last letter, our third letter, A, C, G. Well, all 4 of these actually code for the same amino acid, so it doesn't matter what your third letter is. A, C, G codes for threonine. And so I'm going to put THR. Spelled that wrong. Let's fix that. I just told you I was going to abbreviate it, and I wrote it out. Let me abbreviate it. That's why we don't write them out, because some of them are kind of long. Okay? Let's do our next one, G-A-U. Go to my first letter, G. My second letter is A. My third letter is U. I am coding for aspartic acid, which is ASP. And then finally, we're going to code our fourth one, GUA. Come back here. G is our first letter, U is our second, and A is our third. We're coding for valine. Okay, so we've coded for the amino acids that are needed to build the polypeptide. If this were it, then it would, of course, break off. It would fold up, and you would have your polypeptide or your protein for whatever it is that's being built. There's a couple of things I need to mention, though, here that you don't see because I didn't give you a full genetic sequence for a trait. We have something special called a start codon and some stop codons. So let me explain what that means. Every single polypeptide that's being built is going to have a place to start. When all this is happening, and when the ribosome reads this particular codon, AUG, AUG is the starting point for all of our polypeptides. So if you look at a polypeptide chain, they all start with AUG. That is the met codon. That is the met amino acid. It is the one that starts off all of our polypeptide chains. So anytime this code is read, you know you're starting a new polypeptide. On the other end of that, there's a few that can stop it. There isn't just one that can stop it. UAA, UAG, UGA. These three codons are all considered stop codons. When the ribosome reads those three, they know to stop. It can be any one of these three, but if those are read, the polypeptide chain's done. And so the amino acids will stop building. The ribosome will unhook. There's enzymes that come in and take care of this. And you now have a fully created polypeptide chain. Some of them are really, 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 really long. Some of them are very, very small. But they'll all start with AUG and they'll end with one of these. Okay? So let me kind of review this again because there was a lot that happened here. We start with our DNA strand. We code it to a messenger RNA strand. This is transcription. When this RNA strand is read, and read in sets of 3, it's the same strand that was here. I didn't change anything. When it's read in these sets of 3, that reading is translation, and it is translated into those amino acids with the help of the transfer RNA. That's where they come in. They bring the amino acids by connecting to those codes. Transcription is writing, translation is being read and turned into the amino acids. Of course, once those stop codons are read, then you have the end of this process of protein synthesis, and you have a polypeptide or a protein.